Originally, Rosalind Franklin didn't get the recognition she deserved for her contributions to the discovery of DNA. However, scientists must always build on the knowledge of others before them to discover even greater things. Hi there. Today's lesson is going to be on the structure of DNA. So, DNA and RNA make up the different nucleic acids in our cells. And genes are segments of DNA on a chromosome that code for specific traits. So, our hair color, our eye color, our skin texture, do we have a widow's peak or not? What way does our thumb curve? Are we male or female? All of those different pieces of information. DNA is nucleic acids known as deoxyribonucleic acid, and it contains instructions necessary for a cell to build proteins from amino acids. Ribonucleic acid, or RNA, plays a role in gene expression and protein synthesis. So in 1869, Friedrich Miescher isolated a weakly acidic phosphorus-containing substance from the nuclei of white blood cells, and so he called it nucleic acid. In the early 1900s, Phoebus Levine isolated two different types of nucleic acid. He called them ribose nucleic acid and deoxyribose nucleic acid, and this is based on the sugar that makes them up. Soon after that, Thomas Hunt Morgan provided the first experimental evidence that genes are located on chromosomes, and he did this when he was studying fruit flies. In 1928, Frederick Griffin designed an experiment to study the bacteria that were responsible for a pneumonia epidemic in London. So he discovered that dead pathogenic bacteria passed on their disease-causing properties to live, non-pathogenic bacteria. So they were able to share their material. He called this the transforming principle. Griffith died during World War II, but several scientists continued to build on his work. So in his notes booklet, he had information that he took a living virulent strain from a dead mouse and a living avirulent strain. He injected a mouse with a living virulent strain and the mouse died. He injected a mouse with a living avirulent strain and the mouse lived. He heat killed the virulent strain and the mouse would live. But if he took the living avirulent strain and the heat killed virulent strain, the mouse would die. So from this, he inferred that the heat killed bacteria were able to transfer their virulent properties over to the living avirulent bacteria. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty discovered that when they treated heat-killed pathogenic bacteria with a protein-destroying enzyme, transformation still occurred. So even though they destroyed the proteins, the pathogenic bacteria were still able to pass their virulence on. But when they treated heat-killed pathogenic bacteria with a DNA-destroying enzyme, the transformation did not occur. So this demonstrates that DNA had a role in transformation, that was the carrier of information between cells. So the controls were live encapsulated virulent bacteria and a dead mouse. Live non-encapsulated bacteria, avirulent bacteria, and the mouse would live. We had live encapsulated virulent bacteria and heat-killed virulent bacteria. Then we killed it with heat, the mouse would live. So that was our control. Then for the experiment, live non-encapsulated non-virulent bacteria and a mixture of heat-killed virulent bacteria and live non-virulent, the mouse would die. Then we took live non-encapsulated non-virulent bacteria and encapsulated virulent bacteria. So we've destroyed its DNA, and then the mouse would. So they took DNA isolated from heat-killed virulent bacteria. Then they treated it with, and then the mouse would die. So that live non-virulent bacteria with DNA isolated from the heat-killed bacteria and would still kill the mice. So that that experiment demonstrated that DNA was what carried the information. So continuing on, in 1952, Alfred Hershey and Martha Chase used radioactive labeling to show that genes are made of DNA. So our traits, the information that carried forward from our cells that give us our individual traits, are made of DNA. So they used viruses that contained a protein coat surrounding a length of DNA. And that virus attached to a bacterial cell and injected information into the cell. The infected cell produced new viruses and bursts, which released new viruses and infected other cells. So they were able to attach a virus. They were able to give new information to a bacterial cell. Hershey and Chase created two batches of the virus. One, they labeled the protein coat with radioactive sulfur, and the other, they labeled the DNA with radioactive phosphorus. What they found was the radioactive phosphorus was in the bacterial cells, and that DNA must direct the cell to produce new viruses. So here they have a bacteriophage with the phosphorus labeled DNA versus the phosphorus labeled protein capsulation. But where we have the phosphorus labeled DNA, we found that in the cells, meaning that the phosphorus, it was part of the DNA directing new cell division. After isolating DNA and RNA, Levine determined that both molecules are made up of nucleotides and long chains. And nucleotides are composed of a 5-carbon sugar, deoxyribose in DNA, and ribose in RNA, a phosphate, and one nitrogen base. There's 4 different types. Those 4 nitrogen bases follow 2 different chemical groups, purines and pyrimidines. Purines are adenine and guanine, and pyrimidines are thymine and cytosine. So here is a drawing of the structure of DNA as well as a chemical structure of DNA where we have our phosphate, our sugar, and then our nitrogenous base. So now that we knew that DNA was what transformed cells and controlled their division, we wanted to find the structure of DNA. And in the late 1940s, Erwin Chargaff found that nucleotides are not present in equal amounts of DNA and RNA. They're present in varying amounts. And the number of adenine in DNA is equal to the number of thymine in a sample, and the amount of cytosine is equal to the amount of guanine in a sample. So this constant relationship is called Chargaff's rule. In the early 1950s, a British scientist, Rosalind Franklin, she used X-ray photography to analyze the structure of DNA, and she worked pretty independently. She was able to identify that DNA had a helical structure with 2 regularly repeating patterns. The nitrogenous bases were located on the inside of the structure, and the sugar phosphate backbone is located on the outside, more near the nucleus. Lots of people argue that Rosalind Franklin should have shared in the Nobel Prize for the discovering of structure of DNA, but unfortunately, the Nobel Prize is not handed out posthumously, and she passed away before it was awarded. In 1953, Dr. James Watson and Francis Crick were the first to produce a structural model for DNA. They used a model of DNA closely resembling a twisted ladder that was based on Rosalind Franklin's images. The deoxyribose sugar and the phosphate molecules make up the backbone or the handrails of the ladder, and the paired nitrogenous bases make together the rungs or the steps of the ladder. And these are held together by weak hydrogen bonds because we'll see later when we have cell DNA replication, it needs to break apart and reattach together. And then from Rosalind Franklin's images, they knew the distance between the sugar phosphate handrails remained constant. So her images were essential to their model. So the 2 strands that make up DNA are not identical, they're complementary to each other. So we have complementary base pairs of adenine and thymine and cytosine and guanine. So we have 1 pyrimidine and 1 purine in every pair. And the 2 strands are anti-parallel, so they would be read and replicated in different directions. So each strand of the double-stranded DNA model has a 5' end, and the other end contains 3, and they run in opposite directions. Okay. So here we can see that our base pairs are complementary, our A's and T's complement each other, and our C's and G's complement each other, and that the distance between them all remains constant. So in a segment of DNA, the number of purines equals the number of pyrimidines, and this is because the base pairing rule. So nitrogenous bases always pair with the opposite. So adenine and thymine are constant, and guanine and cytosine are constant. So when we look at this example, if 15% of the bases were thymine, we want to see how many of the bases are going to be adenine and guanine. So thymine pairs with adenine. So we're going to have 15% here. 15% are adenine. So then we have a total of 30% are going to be a combo of the adenine and guanine. So we have a total here. So I'm working with the pen. of 30%, giving us 60% remaining. And if we divide that 60 in two, we're going to have 30% are guanine and 30% are cytosine. Now we're going to look at the complementary strand of DNA. So we have A is going to complement itself with T, thymine with adenine, guanine with cytosine, cytosine with guanine, and so on and so forth. So that complementary strand of DNA is going to remain constant. Adenine is always going to bind with thymine, and cytosine is always going to bond with guanine. So comparing RNA to DNA, the sugar component is different. RNA has a ribose sugar, where DNA has a deoxyribose sugar. RNA does not have the nucleotide thymine, but it's replaced with uracil. So in this case, in RNA, an adenine will bond with uracil instead of thymine. RNA remains single-stranded. This allows it to more easily travel out of the nucleus of the cell. And there are several types of RNA. mRNA for messenger RNA, rRNA for ribose RNA, and tRNA for transfer RNA. We'll talk about these in further lessons. I hope you enjoyed the history and the structure of DNA today. Thanks. Bye.